Um, so, my name's Mark Pallen. I am going to give you 5 lectures. This is the first of my 5 on the general topic of global health and emerging infections. And today's lecture is going to be the global burden of infection and an old foe, malaria. And then tomorrow we're going to look at 2 more old foes, TB and leprosy. So just to give you an overview of how all of this fits together, as I say, I'm going to give you 5 lectures. 2 of them are on old enemies of ours, infections that have been around for a long time, well established. Then we're going to look at the so-called emerging infections, potential for new diseases, new infectious diseases to emerge. Then I'm going to look at eradication of disease and the success that we've had in eradicating infections, particular infections around the globe. Very upbeat talk, I hope. And then, slightly at an angle, but I'm also going to give you a little bit about the lab diagnosis of infectious disease. I am actually a medical microbiologist by training, and I'll give you a clue there, some clues there as to how we actually do stuff in the lab, how we achieve a microbiological diagnosis. Now, I don't like WebCT very much, and I've set myself a task this year of trying to make all my lectures completely open to the world. So this lecture is being recorded at the moment, and it will go up onto Facebook, not onto Facebook, onto YouTube, well, certainly by tomorrow, maybe by the end of today, as a slidecast. And I've set up this Facebook page, which you can just find by going to Facebook, the Bio 303 module, and on there I have posted links already to the slides, which I've put up on a website called SlideShare, so that anyone in the world can see these slides and use them. And I think there was already about 60 people that looked at them the last time I looked. Have any of you looked at them already? No, so it must be all those other people that follow me on Twitter have been looking at them. But they're available for you to download from SlideShare. As I say, you can download this audio-visual presentation. I will try and make a podcast, an MP3 podcast of the lectures, of each lecture as well, and put those up. It's a bit harder to find somewhere to put those, but I'll put those up on the local server. In addition, it's just worth stressing that in the 3rd year, you're supposed to do a little bit of extra reading, external reading, if you want to get a high 2-1 or a first. So on Facebook, I have put up some links to additional material. Sometimes when I do this, people get freaked out. You say, oh, you've given me all these links to do. I've tried to be parsimonious. Don't get freaked out. But if you do want to get a first, then have a look at least at some of that material and at least speed read it and try and get on top of it. Okay, so, and handouts. I will bring handouts tomorrow. This lecture's being produced so up to date. It's kind of red hot with kind of being up to date. In fact, as you'll see later on, there's a paper in there that's only just come out. So I have to prepare the lectures at the last minute. That's my excuse. Also, the office is very busy at the moment. So you will get 2 lots of handouts tomorrow. For today's lecture and tomorrow's lecture will be delivered tomorrow. So you can just make notes as you wish, but you will get the handouts as well. Right, on to the topic of the lecture. So what about infection, the global challenge? So in developed countries, we don't go home to bed at night worrying about the fact you won't wake up because you've got some infection. It's become less of an issue here than it was, say, 100 or 150 years ago. But if we look across the whole world, infectious disease is still a major player. At least 15% and maybe more of all deaths in the world are caused by infection. And even in developed countries, as we'll see in the 3rd lecture, we do see new diseases emerging. West Nile fever, SARS. Germany over the summer was devastated by an outbreak of an E. coli infection that just came out of nowhere. Nobody expected it, nobody was prepared for it. And actually getting control of infectious diseases remains a challenge, as we shall see during the rest of the lectures. So this is some data from the WHO, and it shows you the number of deaths that you get from each kind of disease. And you can look at the whole world, and you can see here things like diarrheal diseases, lower respiratory infections, HIV/AIDS, tuberculosis, featuring there in the top 10 for the whole world. If you go and look at low-income countries, they become even more prominent there. So lower respiratory infections, 2.9 million deaths, diarrheal diseases, 1.8 million deaths, HIV/AIDS 1.5, tuberculosis just under 1 million, and malaria similar kind of figures according to WHO. You get different figures from different authorities, but I guess the WHO is probably most likely to be right. So you can see that really on the world stage these are important players. If you look at high-income countries, then yeah, they do actually disappear off the screen for the most part, apart from lower respiratory infections. And things like the pneumococcus pneumonia is a common cause of death in people who are otherwise debilitated, even in the West. If you have a stroke, you might survive for a year, but then you probably will succumb from a pneumonia, let's say, as the immediate cause of your death. If we look at child deaths by region, again, this is showing you about each of these dark-coloured men here is about 10% of the world's child death due to a specific cause. So you're supposed to kind of read it looking that way. So you can see when we look at all causes, most child deaths are occurring in Africa. We look at diarrheal diseases, the same kind of picture, although there is substantial deaths in Southeast Asia from diarrheal diseases. Similarly with pneumonia, malaria really very highly skewed. Most of the deaths in children due to malaria, and if you look down below, HIV as well, are in Africa. And if we look at adult mortality rates as well in the African region, again we see this tremendous impact of HIV/AIDS and other infectious and parasitic diseases here as major causes of death. Now, as it happens in this series of lectures, I'm not going to cover HIV/AIDS. I could have done, but I had to choose, be selective. But I'm sure from other sources you've already heard something about the problem of HIV/AIDS. Now, the rest of the lecture, I'm going to focus now on one particular problem, malaria, which is a big player on the world stage in terms of both causing death and also causing misery, causing illness, causing lost work time and so forth. So this is, what is it? What is malaria? It's a complex and deadly mosquito-borne infectious disease. It's caused by these protists, what we used to call protozoa, from a particular genus known as Plasmodium. And in humans, there are four main causes of malaria. The most serious kind of malaria, so-called malignant malaria, is caused by Plasmodium falciparum, or falciparum. And then we have three others which cause milder, usually non-fatal disease. These are kind of also-rans. They tend not to kill people. Falciparum is the major killer. These infections are naturally transmitted by this creature here, the female Anopheles mosquito, feeding on human blood, both sticking parasites into the blood in an uninfected individual and sucking them out of an infected individual to be transmitted. Leading cause of morbidity and mortality worldwide, especially in pregnant women and children. About 40%, or just over 40% of the world's population, that's about 3.3 billion people, are at risk of malaria in 109 countries. So it really is a big problem across the world. 250 million cases in 2008 and around 1 million deaths in children is the figure that's usually put around. And as I mentioned before, it's in Africa that we see the highest economic burden. It's been estimated that $12 billion per year are lost in direct losses. So that's the cost of being ill, the treatment, the premature death. But this is also, malaria is also actually a big drag on economic growth. So the fact that people are ill, the fact that they are not working to the most efficiently and so forth, this is a big drag as well. Of course, we come across malaria even in this country. Any of you who watched the X Factor last year will know all about Cheryl Cole's brush with death. She went to Tanzania, she caught malaria. She was actually saying she was sick on the television, and here are some of the things that she said. Now, as you might expect, she thought, first of all, she had a hangover. She thought she'd been drinking vodka the night before, but it got worse and worse. Exhausted, flushes, goosebumps, blue lips, blue fingertips, blue toes, then boiling hot, and then she really was actually in intensive care. She was at death's door. She had no liver function, no kidney function, swallowing fluid, no oxygen in her blood. And she said, you sometimes feel like you say, I feel like I'm dying. She actually felt like she was dying, and she said to the nurse, is this a possibility? And the nurse said, yeah, it is a possibility. She was very close. But luckily for us, she survived, and she doesn't do the X Factor anymore, but she's still around. So what about the details of malaria, the life cycle? Well, this is here to scare you a bit. It's a very complex picture. And we're going to go through each of the individual parts. But this is just to give you the global picture. So there's this cycle in mosquitoes here, and there's these cycles in humans. And we'll now go through that in more detail. So in the mosquito is the primary host of malaria, and it's the transmission vector. It's only the female form of this Anopheles mosquito. And what happens is the young mosquitoes bite humans, and they ingest a form of the parasite known as the gametocyte in the blood meal. Those gametocytes can differentiate into two forms, the male or the female gametes, and then they go into the gut of the mosquito, and they fuse together. Don't have to know all the details of the different stages in the mosquito, really. I'm just putting this up here for completeness sake. But basically, they go through the mosquito, and they eventually end up releasing sporozoites that migrate into the salivary glands of the mosquito, poised to be injected into a human into the human bloodstream when the mosquito next feeds with the saliva of the mosquito. Now, when we get into humans, we have these two major stages, the so-called exoerythrocytic cycle and the erythrocytic cycle. So they go in as an infection there, come in. The form that comes in is known as the sporozoite. So really, you just have to memorize sporozoites, merozoites, and gametocytes, the three main forms you have to know about. Sporozoites go through the circulation, they hit the liver very quickly, and then go into the liver. And once they're in the liver, they then undergo different rounds of differentiation, and eventually they get released as what we call merozoites. And they go through this cycle in the blood, differentiating, rupturing red cells, being released, reinvading red cells, and so forth. And a minority of them differentiate out into the gametocytes, which are what are taken up by the mosquitoes. So just to put that in words, so the sporozoites enter the bloodstream. Within half an hour, they've got into the liver. So there's a very short window of opportunity in terms of targeting the sporozoites as they come in. They get into the liver, they multiply asexually and asymptomatically. The person doesn't even know they've been infected for a week to 2 weeks. Differentiate into these merozoites, thousands and thousands of them, and then the liver cells, the hepatocytes, get ruptured and they come out, these merozoites escape into the blood. Once they get into the blood, as I say, they go through this cycle, the so-called erythrocytic cycle phase. They infect red blood cells and have this asexual reproduction, so they have these so-called ring forms, trophozoites, schizonts, merozoites. Again, you don't really need to know all the details of that, but basically this cycle goes on every 48 hours or so, differentiating, getting to the stage where they then lyse the cells and reinfect. And the burst of lysis and release of parasites actually causes a fever, and that's why we get this cyclical fever in malaria. And as I say, a minority, just some of them, differentiate into the male and female gametocytes. Now there are some, what I've said there is true for all the malarial species, but there are some species-specific features. So in Vivax novale, there are these things called hypnozoites, and these hide in the liver. So instead of going into the liver and then coming straight out into the bloodstream, these actually can hide in the liver, and they can then reactivate after months or years. So this means it's much harder to get rid of the infection in these conditions, and it can grumble on, or you can say get infected, go on holiday to India, and you come back and you're fine and you're fine and fine, but then maybe months later suddenly it reactivates and you get Vivax or Vali infection. Falciparum actually has these adhesins that it produces that get stuck onto the surface of the erythrocyte, and it makes the erythrocytes very sticky, and they will then stick to the walls of small vessels, capillaries, and this causes rupture of the capillaries, hemorrhage, it causes infarction, particularly in two sites, in the placenta and in the brain. And so we see placental and cerebral malaria as complications of falciparum malaria, which we don't tend to see in the others. So where's malaria come from? Well, it appears that it is indeed an ancient enemy of ours, co-evolved with anthropoid ape lineages in Africa. Now, there are some controversies as to the source of human infection, and I'll say more about that in a moment. But once these malarias, particularly falciparum malarias, established in humans, it seems to be carried out of Africa during the out of Africa migration. So everywhere out of Africa was peopled by people from Africa relatively recently, about 60 or 70,000 years ago. If you look in the historical literature, this idea of periodic fevers that come and go every few days have been recorded many times, particularly notably in China back over 4,000 years ago, and it too, in the 2nd century BCE, the Chinese described the Qinghao plant, Artemisia, as a remedy for malaria. And so that was one of those examples where herbal folk remedies were described long ago, and it turned out that this is effective, and in the last 30 years or so it has actually been turned into a highly effective anti-malarial agent. They've distilled out the actual chemicals produced by the plant. Hippocrates described malaria as well. Malaria was carried to the New World by explorers, missionaries, slaves. The Jesuits actually brought back the cinchona bark from Peru, the source of quinine, and again this is another highly potent anti-malarial that's come from plants. As a little story, it's said that Oliver Cromwell had malaria, which he used to get malaria in the UK, in England, particularly in the Fens, and he refused to take this cinchona bark because he said that it was a papist powder, basically because he was a Protestant. He didn't want anything to do with anything that would come from the Catholics, and in fact he continued to be ill, so he sort of paid for his chauvinism in terms of illness. Historically, malaria was associated with so-called miasmas, kind of mists rising up from the marshes, and in fact the name malaria is just the Italian for bad air. And so people recognised that swampy places were not good when it came to malaria, but they didn't have any mechanistic understanding of how the infection occurred, how the disease happened. Just to point out, as I say, that there's some controversies out there about where malaria came from. This is a paper published early last year in PLOS Pathogens saying, well, we can blame Plasmodium falciparum on bonobos. Bonobos, the kind of chimpanzee, sometimes known as the pygmy chimpanzee. And they did some fancy molecular analyses, detected these parasites, looked at the sequences, and came away with the idea, well, that's it. So humans got it from bonobos. In October last year, though, a paper appeared in Nature that said, well, actually, no, that's not true. Humans actually got falciparum malaria from gorillas. And they did a much wider analysis. They looked at over 1,000 different gene sequences from gorillas and chimpanzees, and came to the conclusion that as it says here, one of these lineages from western gorillas comprised parasites that were nearly identical to P. falciparum. In phylogenetic analysis of full-length mitochondrial sequences, human P. falciparum formed a monophyletic lineage with the gorilla parasite radiation. And so basically they say that's the last word on it. I'm not sure. We'll see. Maybe next year there'll be another paper telling us that we get it from chimpanzees again. But I think probably this is actually correct. But it's been interesting just trying to track this down over the last few years. In terms of history, parasites, malarial parasites, were first seen in 1880 by a French army surgeon, Alphonse Laveran, and for that discovery he was awarded one of the first Nobel Prizes in 1907. He suspected, and one of his contemporaries, Patrick Manson, they both suspected that mosquitoes were transmitting the parasites, but they didn't have any direct experimental laboratory evidence for that. That came from this guy here, Ronald Ross. You can see a typical Victorian gentleman. In fact, he was an army surgeon in the Indian Army, and he saw oocysts of falciparum in the stomach tissue of a mosquito, which he had fed on a malarial patient, this guy Hussein Khan, and that was the beginning of a modern understanding of malaria. Actually showed that it was the mosquito. Within a year, he confirmed that mosquitoes were linked to malaria in birds, and he'd actually found the entire life cycle in birds. And for his efforts, Ronald Ross was awarded the 1902 Nobel Prize in Medicine. Ross was a bit of a quirky character, though. He kept these elaborate notebooks describing the ring forms that he saw in the blood and so forth. But he also penned some poetry, couldn't help himself. He actually captured his discoveries in verse here. And he particularly, I like, this famous phrase. He said, seeking his secret deeds with tears and toiling breath, I find thy cunning seeds, O million murdering death. It's a bit overblown, but it actually, I guess it was one of those moments in the history of human progress that was actually worth marking in a big way. But I know this little thing, a myriad men will save, O death, where is thy sting, thy grave, thy victory, O grave. Modern scientists are a bit boring compared to these guys who actually wrote poems about what they did. Anyway, if you listen to the Italians, they'll tell you that Ross was a bit player, and it was these three Italians here, Grassi, Bignami, and Bastianelli, who actually were the ones that first showed that malaria was transmitted by mosquitoes. They were actually the first ones to show that, beyond doubt, that human malaria went through this life cycle in Anopheles mosquitoes. So they obviously do share credit with Ross. And things in those days move very quickly. They showed that if you feed mosquitoes on a patient in Rome and then send those mosquitoes to London and then feed them on volunteers, let them feed on volunteers, those volunteers in London will get malaria. So basically they showed beyond all doubt that it was the mosquitoes that were transmitting malaria. In the 1930s in South Africa, de Mellion showed that malaria could be controlled by frequent spraying of walls and ceilings. And in 1934, a very important anti-malarial drug, chloroquine, was discovered in Germany, and within a decade or more, just over a decade, was shown to be a safe and effective anti-malarial. DDT was shown to be a very good insecticide by this guy Paul Müller, and he won the 1948 Nobel Prize. You can see a lot of Nobel Prizes associated with malaria research, and perhaps justifiably so. And then it took a few years more before people started looking at what goes on in the liver and understanding the liver phase of the life cycle. So what about controlling malaria? What's the history of malaria in terms of control? Well, if we look back at the turn of the 19th century, the dawn of the 20th century, over 3/4 of the world population at that time, a relatively small number, 1.6 billion now, but in those days, but over 75% of the world's population was at risk from malaria, and it was causing over 3 million deaths. And in fact, the major burden of disease was outside sub-Saharan Africa. The first major efforts to actually try and get rid of malaria started in the middle of the 20th century, particularly in the USA. They had the National Malaria Eradication Program. Over 4 million houses were sprayed. And they went from, in 1947, they had 15,000 cases in the continental USA, down to 2,000 within just 3 years. Things were even more impressive in Sardinia, where they went from, in 1947, to 75,000 cases down to 9 cases, particularly using the spraying of houses with DDT. So this led to a call for a global malaria eradication campaign, which ran from the 1950s through to the 1970s. And the hero of this was a guy called Fred Soper. He was an American epidemiologist who worked for the WHO. And this campaign made very heavy use of DDT to spray houses, plus case finding, actually trying to find cases and treating people. And they had this kind of campaign of attack where there's preparation, attack, consolidation and maintenance in terms of their way of trying to get malaria under control. Was it successful? Well, yes, partially. It reduced the world population at risk of malaria from where it had been 75%. It was now down to 50%, around 50% of the world in 1975. And basically countries that had temperate climates like the UK and North America, these countries managed to control malaria, where there was seasonal transmission. They were, in fact, eradicated malaria. Even in tropical countries, there were some spectacular successes. In Sri Lanka, they had 2 million cases or more in 1958, went down to 17 by 1963. But the trouble was that the malaria hadn't been eradicated, hadn't gone away completely. All the conditions were still there for it to bounce back, and it did bounce back with a vengeance in Sri Lanka to 500,000 cases. Okay, not quite as bad as it had been before, but still pretty bad. And the key point about all this, although it was called Global Malaria Eradication Campaign, actually there was negligible progress in many countries around the world: Indonesia, Afghanistan, Haiti, Nicaragua. And in fact, they just ignored most of sub-Saharan Africa as if, oh, that's too difficult a problem. So it's a bit like the US have these World Series where they have the USA and Canada and call it the World Series. You know, a global eradication campaign that ignores sub-Saharan Africa isn't really much of a global eradication campaign. Why did it fail? Well, basically Darwin in the drugstore is one explanation, that if you spray DDT onto mosquitoes, the mosquitoes, by a process of Darwinian evolution, will become resistant. The populations will become resistant. 1 in a billion mosquitoes that show some resistance to DDT will be selected for, and before you know it, you have a population that no longer responds to DDT. Similarly, the use of chloroquine alone led to the emergence of chloroquine resistance in the malarial parasites. Problem also was, the world was not a very safe place in the 1950s, 60s and 70s. There were wars and massive population movements. Donor countries weren't really that interested. They weren't sustaining their funding. It was very much a top-down effort. So, very important. European, Westerners or whatever, or Americans come in, deal with the problem, then go away again, rather than actually getting the local communities involved. And the World Health Assembly abandoned eradication in 1967. They said, this isn't working, let's be realistic, we're not going to eradicate malaria. And the focus was on control, stick your finger in the dike rather than actually trying to get rid of this forever. And in fact, eradication or elimination, these became taboo words in the malaria community. Nobody said the E word for decades. So we're now at a situation where about 40% of the population is at risk. So a little bit better than it had been, but still a big problem. Things went dormant for a couple of decades, well, 2 or 3 decades, and then things started picking up again around the turn of the millennium. This initiative, Roll Back Malaria, was started by the WHO's Director General in 1998 and launched by the WHO with all these other sister organisations. And then there was this Roll Back Malaria Change Initiative coming in. And in 2007, Bill Gates, the guy who made his money from Microsoft, that you will use, Windows that you'll use on your computers, or those of you who aren't Mac users, and even those who are Mac users probably use Microsoft Office. Well, all that money he's actually now ploughing into trying to eradicate these diseases. And he actually, some people argue he's perhaps on the autism spectrum disorder. He sees the world in black and white, very blunt terms. He said, just eradicate. And he mentioned the E word for the first time. He calls for eradication. And he's energized the world, across the world, various bodies, including these global bodies. So the Global Malaria Action Plan hatched in 2008. The idea was that you'd have universal coverage for all populations at risk, with locally appropriate interventions by 2010. They set these ambitious goals of getting down the number of cases by 50% in 2010 and 75% in 2015, reduce malarial deaths by 50% and to near zero preventable deaths in 2015, and eliminate malaria in 8 to 10 countries by 2015 and afterwards in all countries in the pre-elimination phase. Another thing that happened was this global fund was set up. International funding for malaria control went up dramatically from $300 million in 2003 to $1.7 billion in 2009, largely due to the emergence of this global fund and greater commitments from the US President's Malaria Initiative, the World Bank and other agencies. And so there's been an absolutely dramatic scale-up in malaria control interventions in many settings, and this has worked, as we'll see in a short while. So how do we control malaria? What are the options that we have available to us? We have effective medicines and relatively inexpensive preventive measures, but it's hard to roll these out across the world because they're not reaching everyone that needs them, largely because of poverty, but other reasons as well. We've seen new medicines and new approaches developed as well, and the challenge of producing a vaccine that we can use that provides a high level of protection is until last week, or the week before last, is unmet. So we have this issue as well. We'll say more about that, as I say, as a stop press later on. So where are we going to intervene if we're trying to stop malaria from spreading? This just summarizes the whole picture. We're going to go through some of these points in more detail now, but just to give you the big picture, we've got the parasite, the vector, and the host. And what can we do? How can we intervene? So let's look at some of these issues in more details. So we can control the vector, and the aim here is to try and protect individuals against the mosquito bites, as individuals, and at the community level we hope that we can just reduce the intensity of local malaria transmission. And basically there are two options. There are nets and there are sprays. You can use mosquito repellents as well, but those are a minor component. The two big things are using nets and sprays. So nets, we can use insecticide treated nets, and there are this most recent development of so-called long-lasting insecticidal nets. These do not require retreatment, so you can put them out, deploy them, use them, and they will last for at least 3 years. So they provide a physical screen against the mosquito, but because they're actually impregnated with insecticides, they will also kill the mosquitoes if the mosquitoes get too close to them and try and get in there. And in Africa alone, 140 million nets were distributed between 2006 and 2008. Some of you may have watched Cheryl Cole the year before she got malaria. She was involved in an initiative to climbing Kilimanjaro, where they were actually raising money for mosquito nets. So there's a kind of irony that she succumbs to the infection herself the following year. Another option is so-called indoor residual spraying, and I put these malariologists seem to have these funny little acronyms, so they call this IRS. I don't know why they have small letters for the first two and a large one, but anyway, that's what you find in the literature. So it's IRS. So what you do is that you have insecticides and you spray them on the walls of homes. This is a picture from the middle of the 20th century. I think it's in Italy, going around spraying the inside of houses. That was using DDT. DDT then got banned because of its bad effects on the environment. And it's clear that if you go out and spray the environment with DDT, you do lots of damage. But there's been a huge campaign lately, a lot of impetus to actually say, well, we've overreacted, and that DDT in the environment out there is a bad thing, but in homes, used in a targeted way, it actually is the most effective and a very cheap way of controlling mosquitoes in the built environment. Some of the people that are propagating the idea that we should be using DDT and trying to get it off the banned list, they call it a weapon of mass survival, because it really is highly effective when used appropriately. In terms of research, there are various research techniques out there that have been investigated but not yet put into practice. One is the so-called sterile insect technique, where you take lots of male insects and you sterilize them by irradiating them, and then you release them. And they go and mate with the females, competing with the fertile males, and hopefully they just actually bring down the numbers of successfully fertilized males such that the numbers just plummet. It's a bit of a balance. You have to, if you obviously irradiate them too much, they become pretty unfit and they can't find a female. But if you don't irradiate enough, they won't be sterile. But that you can get this right. And in fact, in North America, it's been used on another insect, the screwworm fly, and has actually been very successful in eradicating it. So there's a question about whether this could be used. Another approach, a more sort of high-tech approach, is can we actually make mosquitoes that are themselves resistant to the parasites transgenically? Can we actually give them extra genes, fiddle with their genetic makeup so that they become resistant to parasites? Again, this is something that's been suggested. There's research going on. It's a bit perhaps more controversial than the sterile insect technique because it requires transgenic animals. But there have been some field studies have been done in Australia, and it does appear to be an effective approach. Okay, vector control is one thing. Actually managing cases of malaria effectively is also important, and getting the diagnosis right is important. So the symptoms of malaria is just you just have a fever and you feel unwell. There's nothing very specific about them, and so malaria can be confused with many other different kinds of infection. And so some kind of laboratory diagnosis is really what you need to be able to target treatment properly. And you have a choice. You can either do microscopy, so you can stain up the malarial films. You take some blood and you make a film of it on a slide. You can either make a thin film or a thick film, stain it up with a so-called Giemsa staining technique, and you end up being able to look down a microscope and see this kind of picture here, where you see these ring forms inside the red cells. And this is unusual. You do actually here see a few gametocytes as well, which is unusual. I don't think I've ever seen a gametocyte on one that we've stained ourselves. You can see them in ones that have been prepared by others sometimes. Anyway, it's very cheap, very quick and straightforward, but that's looking at it from one direction. If you look at it from the other direction, though, it does require quite sophisticated equipment. You need good functioning microscopes, and you need well-trained, competent microscopists. And actually finding these, in this case here, where the parasitemia is so high, it's obvious. But spotting the parasites when there's a low parasitemia, it is actually quite an art, and it's something that's not straightforward and trivial. So there's been a lot of interest in rapid diagnostic tests, these so-called immunochromatographic assays, a bit like pregnancy tests for malaria, where you have the strip here and you dip it in to some serum, and out comes a result that you can read. These have all the advantages of that you don't need to train anyone very much. It's quite straightforward, but they are still relatively expensive. And there's kind of lively competition between the two different methods, and there's quite a few reviews. I haven't read every last one of them. comparing the deployment of these different diagnostic approaches out there in the field. Another important aspect of case management is the so-called intermittent preventive treatment for pregnant women. Again, in the malariologist jargon, this is called IPTP. And what you do here, in high transmission settings, pregnant women are given 2 doses of sulfadoxazine pyrimethamine. Regardless, they don't have to have malaria, they don't have to be shown to have malaria, they just get given this prophylactically from the 2nd trimester onwards, preferably 1 month apart. And the idea is to just dampen down the parasite load and prevent the bad sequelae of infection of the placenta, loss of the pregnancy, or damage to the infant such that the infant is very ill at birth. In terms of treatment of patients, we have this twin aims of actually making the patient better, but also preventing transmission, and those 2 things go together. And nowadays, we rely on these so-called artemisinin-based combination therapies. So I mentioned Artemisia, the plant, earlier. These are drugs that have been derived from that plant, and they're used in combination with other agents, and there are a variety of different regimens available, and these are used against Plasmodium falciparum. So this was one of those surprising turns that I think it was in the late 70s, early 80s, people realized that the Chinese herbal medicine did actually work, and in fact it was the most potent thing we had against malaria in terms of treatment. Chloroquine and primaquine against Vivax, and we give prophylaxis to prevent malaria in travellers. Prophylaxis doesn't work on a population-wide scale in areas where malaria is endemic. So what are the problems? Well, we've got some threats here because there is some evidence of resistance even to the artemisinins. Problem is that if people use this drug on its own, one of these drugs on their own, then you breed up resistance. And this is a general kind of rule of thumb for here and in TB and other situations. If you give a single drug, then you can breed up resistance quite quickly. If you give combinations, it's much harder for resistance to arise. And that's just to do with the kind of biomass of the parasite that you have. If you say you've got 1 billion parasites and you've got 1 in 1 billion is resistant in that population, and you give the treatment, then that 1 will become the basis of a new population. If you give 2 drugs, the chances of there being 1 billion times 1 billion parasites in any one individual are just not there. It's not possible. There's just not enough parasites to become resistant to both drugs at the same time. Are there new drugs on the horizon? Well, yes, there are. And in fact, in this area, it's actually, there's a kind of optimism in the air that there's a whole range of new drugs passing. So that when we talk about the deployment of drugs, we talk about preclinical trials, phase 1, phase 2, phase 3, and then approval. And this is kind of regulatory escalator, if you like. You have to first of all show that the drug is safe, can be given to humans. You then have to show that when you give it to a larger number of people, that it's showing some kind of moderate efficacy, but also continues to be safe. Then you can compare it with existing treatments and say, actually, it is as effective as what we currently have, or it's better. And so there's a whole range of these that are being developed. In some cases, there are combinations of existing drugs that are already known about, we already have, that are actually moving into trials. You see, so this one here, azithromycin and chloroquine given together. Azithromycin has been very widely used as an antibiotic against bacteria, but it appears to have anti-malarial activity, and if you combine it with chloroquine, it does appear to work. So there's a great deal of hope there, and there's an organisation called Medicines for Malaria Venture, which is really pushing this forward and driving the research agenda. If we want to develop a vaccine, what about that? Well, it's hard to get a vaccine against the blood stage, the marozoites and so forth, because there were just so many, there are billions and billions of these parasites, huge biomass circulating in the bloodstream. And to expect the immune system to be able to eradicate all of those is asking too much. Particularly as the parasite actually has these very wily mechanisms in terms of changing the proteins on its surface such that it can evade the immune response as well. So it's going to be, this is very hard. Most people, don't think this is going to be possible. Although it is true that if you go to an endemic area, people get repeated parasitemias, and they do develop some degree of immunity that actually stops them from getting clinically ill. And in fact, if you have someone, say, who's brought up in Africa, they live in Africa during their childhood, and then they leave Africa and they come to the UK, sometimes they can then relapse with parasitemia because they can get reinfected and then end up getting very ill when they think, oh well, I never had this trouble when I was in Africa, and that's because in those endemic areas they're continually getting, if you like, a booster vaccination, and when that's taken away, they then lose that immunity. So it's quite short-lived immunity anyway. So basically there are two options that are being looked at. One is looking at pre-erythrocytic parasite stages, looking at the sporozoites and targeting a particular antigen, the circulating sporozoite antigen. And these subunit vaccines are actually now in phase 3 trials, this so-called vaccine, this RTSSS vaccine. Another option is that you could actually target the gametocytes. Now the gametocytes are the things that the mosquito is just about to suck up and get infected with, and then the mosquito gets infected and then infects someone else. These so-called transmission blocking vaccines are kind of, ethically, they're a bit of an oddity because they're not actually helping the person you're vaccinating. That person's got malaria, still going to have malaria. But what they're doing is preventing that person being able to spread malaria because the infectious forms, the gametocytes, are actually being destroyed. And so there's a lot of lively debate about whether those vaccines could be used, should be used, and so forth. But in the last few weeks, there's been a great deal of interest actually in these pre-erythrocytic vaccines. And just was it last week or the week before, we had this in the New England Journal of Medicine, first results of a phase 3 trial of this RTSS malaria vaccine in African children. And this is the first time that we've really had anything that looks like an effective vaccine against malaria. Unfortunately, it's not perfect by any means. It was only giving about 50% protection in this study. So, it depends if you're a cup half full, cup half empty person. 50% is actually a lot better than anything else that's ever been achieved in the history of this kind of research. But it still falls short of being the perfect malarial vaccine. But at least it gives us hope. And there is a lot of discussion now as to how this is going to be rolled out. There's going to be more trial data coming in. It's likely the WHO is likely to recommend, if things continue to be looking good like this, recommend this as a routine vaccine and put into routine use. So in the last couple of minutes, are we winning the battle against malaria? Well, I stole this from an article in Science. It came out last year. We look at the countries that are reducing the number of malaria cases between 2000 and 2008. There are many countries where there's been a decrease in cases of greater than 50%. So Botswana, Cape Verde, Eritrea, Namibia, Rwanda, Sao Tome and Principe, South Africa, Swaziland, Zambia. All of these countries have seen malaria rates plummet below 50% in that period. Unfortunately, and if you look elsewhere in the world, you see similar kinds of progress. Lots of in the Americas, Southeast Asia, European regions, everywhere we're seeing good progress. In India, we've seen decrease, not quite as dramatic, but still 25% or so. But there are some still problems out there. There are some countries, particularly in Africa, where there has been limited evidence of decrease at all, and a rather depressing list of countries there. And again, this comes from the same article, which I've put a link to on the Facebook site. If you look around Africa, you see a very mixed picture of what's going on. So places like Rwanda, malaria down more than 50%. Similarly, Zambia, malaria deaths down 65%. But there are a few hard problems. This is the hardest, I think, of all. Democratic Republic of Congo. It's a country that suffers terribly from war. 5 million people have died from wars in the last few years there. But even so, this is number one killer here, malaria. But as I say, 12 million bed nets have been distributed, even in the Democratic Republic of Congo. So, will we win? I think I think the consensus is that the complete interruption of malaria transition, we're going to need new tools. We're going to need that new vaccine. We're going to need other ways of dealing with it. We're actually in a precarious situation because we're relying on these artemisinin derivatives and these pyrethroids that are used for spraying currently. And if resistance to those becomes widespread, then we are losing our frontline agents. Now, as we said, there are drugs coming up behind, but what if they don't pan out? So we really do need new tools, other preventive measures, diagnosis, treatment, surveillance. And I put this down here. This is my own personal view. I mean, we need places like the Democratic Republic of Congo to be at peace. I mean, it's impossible to go out and deploy public health in a war zone, and so we need, both to decrease the number of deaths from warfare, and also obviously economic development is important as well in those situations. We'll be seeing later on that in another disease that we're going to deal with, guinea worm, actually it has been a problem in Sudan, and particularly in southern Sudan, because of the war. And now that the war is over, things are moving forward very quickly, and hopefully we can see the same with malaria. Okay, so will we eradicate malaria? I don't think we will in my lifetime. I'm getting a bit old, middle-aged. Well, I'm getting middle-aged now, and it would be nice, but I don't think it's going to happen. It may happen in your lifetimes. You're going to be 30 years younger than me. It may happen, or in your. I think it would be nice to think that really it will happen in your children's lifetimes. And just, I think, we've just about got time. I've got a little film I was going to show you just to finish up. This is about the Global Fund and the rationale for it.
In fact, I called it a war chest. Quite a lot of people laughed it off, saying, "There he goes again, dreaming." And I love dreams. It always starts with a dream.
Ask yourself, what do you need to live? Food? A home? A job? Your family? Love? And health? Without health, the others are not enough. It's one thing we all have in common.
If we are in the same boat, then a hole at one end of the boat puts all of us at risk.
Around the world, AIDS, tuberculosis, and malaria kill millions of people each year and handicap the development of entire nations. These diseases are not invincible, far from it. But to fight them, it takes commitment, the right strategies, money, and leadership.
The creation of the Global Fund was one of the kindest, most important things that happened to help improve people's lives. The treatment, whether it's for tuberculosis or AIDS or malaria, is saving lives, and that has a catalytic effect on allowing the community to take care of itself.
In 2002, the Global Fund to Fight AIDS, Tuberculosis and Malaria was founded to bring massive amounts of new funding to the fight against these three diseases.
There's truly an era prior to the Global Fund and an era after the Global Fund. When it comes to AIDS, in 2001, 2002, there was barely anyone on treatment in the developing world. Today, it is over 4 million people who access antiretroviral treatment, the life-saving therapy for AIDS.
2/3 of all international funding to fight TB and malaria, and nearly a quarter of the money used to fight AIDS, is now being channeled through the Global Fund.
I think it's fantastic to see how money provided by donors turns into real help, real medicine, and save the life of real people.
Through the Global Fund to fight AIDS, tuberculosis and malaria, over 3,500 lives are saved every day. That's 1.3 million people every year. We can win the fight against these diseases. It's just a matter of galvanizing the funding and getting the political will to achieve this.
Today, hundreds of thousands of health workers around the world are dedicating their work to saving lives with resources channeled through the Global Fund.
It's the countries that are in the driving seat. The countries decide on their programs, they decide on their budgets, they decide on how the money will be channeled. What we ask in return are the results.
The Global Fund is flexible. It doesn't make a one-size-fits-all recommendations. The fund's focus on results has really helped drive progress towards the Millennium Development Goals.
While making sure that donor resources are being put to good use, the Global Fund is also strengthening ways in which partners can work together to fight diseases.
No one can fight an epidemic alone, nor the Ministry of Health or the public sector or the private sector or the civil society or the international organizations. When it comes to fighting an epidemic, everyone has to come together.
Well, the Global Fund is run very professionally. It sets big goals, it measures itself, it's constantly improving, and the injection of resources, expertise from businesses could help the Global Fund be even more ambitious.
All of us can be proud of what we have established, what we have done. But more importantly, be conscious of what still needs to be done.
There can be no let up in the fight against AIDS, tuberculosis, and malaria. If we ease up, these diseases will re-emerge. We either win this fight or we lose it. The success of the Global Fund is a test of global solidarity.
If we are to achieve the goal of eliminating transmission of the virus from mothers to children by 2015, there is still a lot of work that has to be done, but it is within our reach.
We can turn the tide on TB and ensuring that we've halted and begun to reverse the incidence of TB by providing access to treatment to those who need it. We also have the opportunity to end malaria deaths. Just like HIV, AIDS, and TB, we have the tools needed to prevent and treat malaria. Death by a mosquito bite, no. Not in the 21st century, we're not having that. These are very big and very bold and ambitious goals, but they're absolutely achievable.
Okay, I think it's worth watching that just for that last quote there by Bono, death by mosquito. I mean, it seems a bit absurd that people are still dying from mosquito bites. Okay, I think we ought to leave soon. If there's any quick questions, I'm happy to take them. Otherwise, I shall see you all tomorrow. I'll be talking about tuberculosis and leprosy.